The biological engine driving insect survival is right there in their phylum name, Arthropoda. It literally translates to jointed legs. For true insects, locomotion relies on exactly 3 pairs of these jointed appendages. They originate exclusively in the thoracic region, anchoring to the body along a seam known as the pleural line. This handwritten class note highlights a critical distinction for your exam. Any leg-like structure attached outside of the thorax is a pseudo-leg or false leg. True legs are strictly thoracic. Moving down the page, we see the absolute baseline anatomical formula. The note here states that accurately drawing and labeling these basic parts is worth 50% of the marks or leg-related questions. You have to memorize this exact sequence: coxa, trochanter, femora, tibia, tarsa. Let's map that formula to the diagram. The coxa stands for the coxa, the sturdy basal segment attaching directly to the pleural line. The T is the trochanter. It acts as a small, specialized hinge joint connecting the coxa to the leg. Next are the primary extensions, the FE, or femur, the largest and most heavily muscled segment, followed by the slender TI, the tibia. Finally, we have the terminal segments, the TA, or tarsus, with multiple small joints, and the pretarsus. Every highly specialized leg you encounter in entomology is simply a structural modification of this exact 6-part blueprint. Looking further down the notes, we see how insects adapt this blueprint. Unmodified legs used for standard walking are called ambulatory, like on a cockroach. When the femur and tibia elongate for running, like in the tiger beetle, it's called a cursorial leg. For jumping insects, like grasshoppers, we see the saltorial type. The defining feature here is a greatly elongated, heavily muscled, and robust femur to generate launch power. This next page diagrams the raptorial, or grasping leg, of the praying mantis. Notice how the coxa is heavily elongated for reach, while the femur and tibia are lined with sharp spines to trap prey. Below that is the fossorial leg, used for digging by insects like the mole cricket. The key modification is the tibia, which flattens out into a heavy, rake-like structure to move soil. In aquatic environments, water scavenger beetles use a natatorial, or swimming leg. The tarsus is heavily modified, bearing numerous fine hairs that act like the blade of an oar. Moving to page 4, we find the forageal leg of the honeybee. Here, the first segment of the tarsus expands to form a specialized pollen-carrying basket. Finally, parasites like the louse use a scansorial, or clinging leg. The tarsus is reduced to a single segment equipped with a sharp hook to grip hair shafts. For exam purposes, you must link the specific functional name to both the insect example and the exact anatomical segment that was modified. Let's move from walking to flying. Insect wings are adult outgrowths of the exoskeleton, utilizing thin membranes supported by a network of rigid veins. According to this section of the notes, wings are never located on the first thoracic segment. They are found strictly on the second and third segments, which together form the wing-bearing region known as the pterothorax.
While flight is the primary purpose, these structures serve 7 distinct functions. They help insects search for food, find their mates, locate resting places, escape enemies, or produce sound, act as an organ of equilibrium, and fan beehives in the summer. To study how a wing operates mechanically, entomologists divide its surface into 2 main geographic regions, the remigium and the vanal area. The anterior portion is the remigium. This is the stiffest part, packed with the highest number of longitudinal veins, providing primary aerodynamic lift. Behind that lies the vanal area, or the clavus in the forewing. Here, the structural veins are much more sparsely distributed, allowing the membrane to flex. Tucked at the very base, closest to the insect's body, is a small lobe called the jugum. It is separated from the main surface by a crease known as the jugal fold. Understanding these surface divisions gives you the geographic map needed to track wing venation and structural changes across different insect orders. A typical insect wing is modeled as a triangle with 3 margins and angles. The front is the costal margin, the outer is the apical, and the lower is the anal. They meet at the humeral, apical, and anal angles, respectively. Inside that triangle is the wing venation. The arrangement of these veins acts as a structural skeleton and follows a standardized nomenclature known as the Comstock and Needham system.
The system categorizes the skeleton into two types: longitudinal veins that run from the root out toward the margins, and smaller cross veins that connect them laterally. This page lists the high-yield longitudinal veins, starting from the top: costa, subcosta, radius, median, cubitus, and anal veins. The key cross veins bridging them include the humeral, radiomedial, and mediocubital. The precise placement and density of these veins within the triangular margins dictate exactly how rigid or flexible the wing will be during flight. In beetles, forewings undergo hardening to form protective sheaths called elytra. Grasshoppers have tegmina. True bugs exhibit hemelytra, split into a hard basal corium and a membranous clavus. In flies, hindwings reduce into small knob-like helters, acting as gyroscopic balancing organs. For insects with two full pairs of flying wings, flying efficiently requires both pairs to move in perfect synchronization. To achieve this, they use wing coupling mechanisms to lock the fore and hind wings together. Wasps and bees achieve this using hamuli. As shown in this diagram, these are rows of tiny, curved hooks on the leading edge of the hindwing that snap into an upward fold on the forewing's trailing edge. Other insects use different mechanical locks. Some use a frenulum, a bristle on the hindwing hooking into a catch on the forewing. Butterflies rely on the amplexiform method, where the margins of the two wings simply broadly overlap each other to move as a single sheet. To ace your ENT 101 exam, you must be able to recall these specific pairings. Practice sketching the 6 basic leg segments of the coxa-tarsus sequence and commit the 3 margins of the typical insect wing to memory. Practice these diagrams accurately